Hello students, how are you all? I'm sure that you are doing good. And this chapter is Principle of Inheritance and Variation. Actually, you are well aware of the principles of inheritance and the variation because similar characters are being transferred or transmitted from parents to offsprings. That is why all the animals and human beings look similar, but not identical because of the variation which is taking place, maybe due to some errors in the DNA duplication, and because of mutation also taking place sometimes. So that is why we are not identical. Generally, if identical twins are there, that different reasons are there for them. You might have studied that also. But here we are to study about the inheritance, the laws of inheritance and variation. So, what is the science behind? And the father of this science, which is known as genetics, is Gregor Mendel. He worked with pea plants because the pea plants were available in abundance, and also pea flowers are bisexual, and then generations of pea plants are very short. So he could study the pea plants generation after generations conveniently. So over the years, he worked with these pea plants and took seven contrasting traits, characters and traits, okay, and tried to derive at some laws, okay. So those contrasting traits must be studied, must be read in your books also. So they are like the tallness, dwarfness, okay, the violet or white color, and the uh, position of flower, there is axial or terminal, and then the pod color, green or yellow, and the seed shape, rounder, wrinkled, and also seed color, yellow, green. So these were the seven contrasting traits that he took. Now, the what he did was he for example in a experiment with two with a pair of contrasting traits, which is called a monohybrid cross. When one pair is there, it is called monohybrid. When it is two pairs, it is called a dihybrid. So in the monohybrid cross of tallness and dwarfness, he crossed in the parental generation. So this is shown as capital T because this tallness is dominant, and the alleles are always in pairs. So it is shown in pair. Capital T, pair of capital T into a pair of small t, which is recessive or hidden. So in F1 generation, what he observed, what he found, if you draw a diagram which is called the Punnett square, right? And if you put these gametes here at the top and at the side, at the top, the dominant pair, that is capital T, in the side, on the side, it is the recessive pair, that is small t, small t. Now this will combine with, all will combine, and this small t will combine with capital T, result will be small t, capital T. Here also you see this small t is combining with this capital T, this gamete with this gamete. So what is the result? The small t and capital T, okay. Here also this gamete will combine with the gamete at the top, so small t, capital T, okay, one recessive, one dominant. Here also it will combine with the capital T. So what is obtained? It is the small t, capital T. So all these four hybrids are containing both the genes of tallness and dwarfness. This is clear from here. That means the dominant trait, which was of tallness, actually dominated, and all the four plants are tall. Though they are having both the genes, they are having the genes of dwarfness also, but that has not appeared. Only the trait which was dominant has appeared in F1 generation. So this is the law of dominance, he said. What he said? Law of dominance. Number one, law of dominance. So what he found here is the law of dominance. He framed this law as law of dominance because only one trait which was dominant appeared in only four plants, the progeny of F1 generation. Okay. So symbolically also you can draw it in a diagram which is called the Punnett square, and then what he did, he self-pollinated all these four F1 plants. Okay, all the tall plants, because all appeared tall, though they were having the genes of the dwarfness also. But in after self-pollination, what he found in the F2 generation, here you can write it like this in this square. At the top, both the genes are there in the self-pollination in the plants, capital T, small t. This is the dominant one, this is recessive. Here also, in the side also, the gametes are one recessive, one dominant. So when they are self-pollinating, what is happening? Small t, capital T. Here it is small t combining with capital T. Again, small t is combining with small t, so it is a pair of small t. And capital T is combining with capital T, so it's a pair of capital T. And capital T is combining with small t, so it is a pair of capital T and small t. So what is found here? One pure tall. This is one. This is the genotypic ratio. The genetic constitution. What you find, what you see here, genetically, as far as the genes are concerned. Both the genes are there. This is pure tallness genes are there. So this is one. It's always in pair. It is the genes of tallness. So one is there. And hybrid genes, hybrid plants having both the genes T and T, small t and capital T are 2. So it is 2 in the middle. And again pure dwarf 1 is there. Both the genes are of dwarfness. So it is pure dwarf. Okay, so it is hidden, so in a small letter, so it is again 1. So what is the genotypic ratio here? 1:2:1. So what has happened here is the recessive character has also featured, the hidden trait has also featured in F2, maybe in the ratio of 3:1. So in all the 4 plants, 3 plants are tall and 1 is short. So ratio is 3:1, that we can see from outside. That is called the phenotype. Now, what is the genotypic ratio? What is the constitution of the genes here in the 4 plants? So you can derive at a similar way as I just now showed how you can arrive at these combinations. Okay, in this Punnett square, which is showing that character is segregated here. So that is the law of segregation. The hidden trait has segregated in the F2 generation after the self-pollination of the F1 plants. Phenotypically in the ratio of 3:1 and genotypically in the ratio of 1:2:1. Clear? Law of segregation. Right, children? And the third law he derived from a dihybrid cross, where he took two pairs of contrasting characters. Here it is only one pair of contrasting traits or characters. There he took dihybrid cross, 2 pairs of contrasting characters. Supposing seeds of the seed, shape and color, round yellow and wrinkled green. These pairs of 2 pairs of contrasting traits he took in this experiment and derived at the third law, which is the law of independent assortment. That is, all the progenies, all the 4 plants derived after the cross pollination of these in the dihybrid cross, and after the self pollination. In the first cross, the dominant pair will be there, will be more. But after self pollination, in the F2 generation, there, what you get 9 is to 3 is to 3 is to 1. That is, all the characters are available in the in the progeny. All the characters are available in the progeny, irrespective of the pair they belong to. So that is the law of independent assortment. Okay, so that I will explain in my next video, students. I am ending this here only, and there is another law, that is law 3, that is law of independent assortment. Okay, so that I will explain in the Punnett square in my next video. Okay. Independent assortment. So with this I am ending here this video, students. I think I will, in my next video I will explain the other important points also of this chapter, and the third law, that is the law of independent assortment. Okay, so God bless you all. Thank you very much.